Welcome you all to this cryo-EM course. Last class I already discussed about the lipid protein interaction, DNA protein interactions, and then I also discussed how to image the bacterial cell and then any kind of other cell, so even fungus also, that you can use the similar approaches, but there is no some cases it will vary based on your sample to sample. So there is no strict protocol or recipe that I can tell and you can mix and match and you can get a fantastic results. For this negative staining, always it is practice. You just have to do these things again and again. Sometime you, based on your sample, buffer condition, staining solution, microscope condition, you have to optimize some parameters and then you can get a good image. So those things you have to play around, and that will be helpful for anyone. So sometimes this practice is very important. If you do more and more and more negative staining, and if you prepare this sample again and again, your hand will be better and you can get a much better data. And also if you do a mistake, if you find out that okay, your staining is not good and your sample is not clearly visible, you have to play around with the different condition and you learn from your mistake, and then it will help you to improve your staining condition. Now today I am going to talk about little bit different things, that is called the sectioning using some kind of ultramicrotome. Here we are just using like a UC7 ultramicrotome, because that is the facility available in our facility, and there are many places people are using this ultramicrotome, or it is called cryo ultramicrotome, to do the sectioning. What is the meaning of the sectioning? That okay, so far we discuss about the purified protein sample or purified DNA sample, purified RNA sample. What about, there are many groups are working, they are using a tissue sample or they are using a lot of cell. Here I showed you that okay, there is a isolated cell, that isolated cell is interacting with the other protein or other cell or some kind of inhibitory peptides or drugs, and then we are imaging. What about when there is a huge amount of cells are growing, and then all the cells after centrifugation, all the cells will precipitate, and then we are taking that precipitate for the next step or next study. So in this particular case, generally we are using ultramicrotome. Not only that, using, so we have a bulk amount of cell, and in that cell we want to see inside that cell what do you have there, or we have a tissue sample. So then inside that tissue we want to see the morphology of some organelles, so something like that mitochondria or nucleosome or ribosome, we want to see it. Then what we have to do, somehow either we have to rupture the cell. Whenever there is a rupture, so then what will happen, most of the things will come out and spread all over. So they will be not located in one particular position. But if I have a cell, if you just consider this is a cell, and if there is a way that I can cut this cell into two species. So if this is the cell, and if I cut this cell somehow and open it up, I can see what do you have inside. So if you look at this cell, that you just consider this is one some component inside the cell, this is another component, but when there is an intact cell you cannot see it. So somehow you have to open up. So we do not want to rupture the cell membrane. When you will rupture the cell membrane, everything will be spreaded all over, those things will be not located in one particular position. But if we somehow remove this outer membrane, if we just somehow open this, so what we can see, we can see all the organelles here. So that is basically we are doing using ultramicrotome. So here if you look at this particular image, you can see that okay, this is the instrument which is called the ultramicrotome. So you can look at that and anyone can tell, oh, it is looking like a microscope, right? You have some to binocular, you can look through this, but basically not. So this is a instrument which we can use it for sectioning the cell, and this is the control panel where we can use all the parameters to regulate this ultramicrotome. So here I am just going to show you some of this component of this ultramicrotome, which it will help you in the later stage if you want to use this instrument. So first one, as I mentioned, it is the binocular. Binocular is located here. Then we have the magnification knob, then we have the focus knob. So here we have the focus knob which we are using to focusing the sample. So why do we need this? Because even if you have a very small tissue, I will show you how to cut that tissue, or what kind of principle we should follow before cutting that tissue properly. So here, so those things are required, that okay, we have a microscope attached here where we have the binocular, we have the magnification knob and focus knob to look at the tissue or the cell pellet properly before doing the section, because this microscope, it will help you to enlarge those portion and then you can use it and do the sectioning properly. So now here you have a knob to tilt binoculars. So sometime you have here you have the, this is the foot, this is the knob which you can use it to tilt the sample, because here we are talking about to whenever we will do the sectioning, we have a tissue sample. That tissue sample, it will come and float on top of a water, and that you can adjust how to that you can use this knob, that knob number 4, to adjust that one. So whenever we are talking about a tissue sample, so there will be a instrument like that, so where we have the tissue will be attached here, a small piece of tissue or a small piece of cell pellet, it will be attached here, and that we are using to section. This is the area we are going to use and section this one. Then here knob to move the binoculars. So knob number 5, you can see it, that is move binoculars forward and backward. If you just want to go back and forth, that you can use this particular knob. And this is the most important knob, that is the cutting wheel. So this cutting wheel, that you will rotate this cutting wheel constantly in one particular direction, and the sample, it will go up and down like that. And there will be a knife, so glass knife it will be there, so your sample, it will stay like this, and there will be a glass knife which one is going to cut this particular area, and this wheel, it will rotate like that, and this sample, it will go this sample, it will just start rotating up and down like so, and then you will get a small section of the cell. Now here, that 7, that is the stage with the knife holder. So this is the number 7, which is the stage with the knife holder. Then 8 is the cantilever arm, where we are going to put our tissue sample here, and the knife, it will be located somewhere here, and which is going to help to cut the sample. So the cantilever arm with the chalk and the segment arc, this is the portion, and the 9 is the Allen key to adjust many things. So now I am going to the next one, where you can see all the details here. So I am just focusing on this, this is the same ultramicrotome, and I am just focusing on in this particular area where you have the sample holder area and your knife holder area. So here, this is the stage, guides with knife holder, and here this is the cantilever arm where we have the tissue sample, and the segment arc which is helping to cut the sample, and this is the cutting wheel from the side. This is the same cutting wheel, you can see it here, which will rotate in one particular direction, and your sample will just go up and down to give you the small pieces of the sample. Now this is basically knife holder we are trying to insert here, which is helping. So your knife, it will located here, your sample, it will locate here. So this one, the one, it is the cantilever arm, then 2 is the, here you can see the 2, which is the cantilever arm set screw, where you can set the screw and your sample, that you have to secure your sample here. So whatever that resin block you are going to fit it, or your solid sample or tissue sample you are going to fit it, it should go and fit it here somewhere, and this screw, it will help you to secure that, so it should not move and it will fix properly. Okay, so the next one is the, so there are some other parameters are there where you have to fit your glass knife here, and then different types of angle that you want to during the cutting, you have to move your glass knife at a different position, different angle, that you can use those screw to adjust those glass knife at a different position. And this is another screw, that is that knife carrier pivot or tilt control knob. So this will help you to control the knife carrier pivot point or the tilt controller. Now if we go to the next one, you can see this is the area where we have the sample, that is basically telling that okay, this is the holding the specimen block. So specimen block means that you have a epoxy resin or solidifying resin block, where you have the tissue sample will be embedded in this resin block. Now what you have to do, this is the resin block, it will be very difficult to cut that sample. So somehow you have to take those resin properly. So you have to look at those sample, that block, that resin block or epoxy block, and then you have to remove those extra portion, that plastic hard material portion, and this is the soft tissue. So you have to make that tissue in such a way like a pencil tip. So then this is the area your tissue will be located, and you will just focus to cut that tissue only, not anything else. So similarly there are some other parts are also there to holding that tissue as well as tightening that tissue, and then fix that entire setup in one particular position. So it will help us to cut that sample properly. So here if you just go through one by one, that is the knob that rotate the sample, the knob that tilts the along with the segment arc. So that will, if you want to, so if you want to move that your sample, or if you want to move or tilt that sample in one particular angle, that time these two particular knob, that number 3 and number 4, it is going to play an important role here. And then trimming block. So that is the 5 is the trimming block, where you are going to do the trimming and you will get that sample. Now at the end we are showing you here that whatever I explained so far. So this is the that entire sample holder unit, where this is the sample where you have a resin block and the tissue is located here. And you can see this is a glass knife, that this is the glass knife we are talking about, and this glass knife most of the time it will be a this kind of shape, will get it, and this is the edge which is going to cut that sample, and this is the area where you have a small amount water which is going to store that different section, and this glass knife is going to help us to cut the sample properly. And there are lots of other knobs are there which is helping us to rotate, hold the sample properly, and then tilt or move in a different position, and then we need that controller to move and cut that sample precisely. Now this is the basically normal diagram how we can make that sample ready. So here if you look at this particular block where we have a tissue sample here. When we have this block, those are some kind of resin which is solidifying material and fixing the tissue. So, when this entire thing is fixed here and the tissue is somewhere here. So, as I mentioned that the tissue area is, if you just consider this is a pencil and this is the tip of the pencil, somehow we have to remove most of this plastic nearby area, or in the pencil most of the wooden part from this nearby area. So, what we need a blade and first gradually cut this top part. So, this entire thing will be very smooth and very smooth, and then gradually you have to remove most of this plastic how we are sharpening our pencil. So, here we are using a blade and then we are trying to remove this plastics area, and we are making a tissue face is a trapezoid face. So, which will only focus on the that particular tissue, and this is the block. This particular block it will go and it will fit somewhere here, and this is the tissue area which is looking towards this glass knife, which will help us to cut that sample. Now, this is how we make that sample ready. So, initially specimen blocks are trimmed to expose the specimen and to form a trapezoidal cutting face. Usually a single edged razor blade is used for this step, which is done by hand while observing the process under the dissecting or stereo microscope. Nowadays, that automatic trimmers are also available, that we can use that automatic trimmer and trim that rest of the area and only focus on this particular sample. Now, this is the obtaining the knife. So, there are two different types of knife we can use it. We can use the glass knife or we can use the diamond knife. So, diamond knife will be here, which is cutting, and this is the area where it is going to store the section. Now, if you look at this, this is the glass knife what I was drawing in the last section. If we just go here, you can see the overall shape will be a triangular shape. So, here if you look at, this is that glass knife where this is the edge. This edge should be very sharp. If it is blunt end, then it is going to damage your tissue sample. So, we are using this glass knife to cut the section, and when that is ready, this is how we are doing that section. So, this is our specimen block where the specimen arm it is going to hold this one. We have to tighten this entire thing, and if required we can tilt or you can change the position of the tissue sample. There are multiple screws there in the ultramicroscope, multiple buttons are there which we can use to regulate the position of this tissue sample. And then this is the knife. We are using this knife to cut this section, and it will store in this liquid water, in the water. So, here we have the specimen block. This is the glass knife again, which is making this very small section and which will float on top of this water. Later on we have to collect those tissue sample on top of the grid, and then we can use this one for other purposes. Now, as I mentioned that so there are couple of important part that we should all focus on that. So, when there is a if this is our tissue sample, if you just consider this is our tissue sample and we want to cut this tissue sample, and if this is the edge of the knife, how our tissue sample and knife should be positioned, because as I mentioned that there is a multiple buttons and handles are there that you can regulate the position of the knife or tilt your sample or position your sample accordingly. So, now if your knife is tilted and you are trying to cut this sample, definitely it will not give you the best data. So, this is the incorrect position. So, your tissue and your knife should be located in this pattern. It should not be in this pattern. If this tissue surface is smooth, if there is no tilt in the tissue sample. Now, if the tissue sample is somewhere tilted, if it is not parallel like that, in that particular position you should tilt your blade accordingly. So, you can get a this kind of shape of that section. So, we always follow this protocol to make our blade as well as sample, and we modify both the things accordingly. Okay, there are couple of more suggestion that okay, if your sample is tilted like that and if your knife or blade is going to do a section, most of the time this kind of blade position, and if your tissue sample is located like that, it is going to break the entire tissue, because it cannot apply the force properly, or sometime it will break or damage the tissue, something like that. This is the small part will come out from that tissue. So, always your tissue sample will be located like this and your knife will be parallel with this one. So, you can get the proper section of that particular tissue. So, if we just go, this is from the top how your sample should look like. If this is your tissue sample, here you can see that okay, this is the resin or plastic block where your tissue is embedded. Now you have to remove that. First, at the top you should do a section and make that surface as smooth as possible. So, from top the sample should look like this at the beginning. So, this is your tissue sample and this is the plastic resin block, and your tissue is embedded within that block. Now, you use a blade or automatic system that one side of that plastic block you should remove it as much as possible and make a shape something like that. So, then we will target the other side and remove the other side also. Now, in this particular situation, if you look at the sample from the top, what you can see, this particular position, it just remove certain area. Then when you will remove the other part of that plastic, so you are just remove in this particular area at this moment. So, this one is the first one. Then C is correlated when you are removing this particular part as well as this particular part. So, you can see both the side tissue is embedded here. The similar protocol, you will just rotate that block, and the other side means this particular side and this particular side also you will remove. When you will remove all these four sides, you can see now the sample is look like that. This is the tissue area, and then you have the four sided cutting, and it is looking like the tip of that pencil. So, now you can use this particular area to do the cross section. So, this is the automatic method that you can use it. So, we will just go and discuss about that automated method, how we can do this sectioning. Before that, we just want to go through a couple of important points, that is the chemical fixation of the sample. So, if we are to study the location of a molecule within cell, then some form of the immobilization must be taken place before sectioning or so, we need some kind of sectioning and fix that cell that I already discussed. For example, molecules that are not immobilized may be displaced from their normal location, resulting in another either a false localization or a negative result, such as when molecules are washed away. The easiest immobilization methods is by chemical fixation using aldehydes to cross-link cellular components. In addition to immobilizing the antigens in place, fixative also help to preserve specimen's morphology. So, we can, for the immunochemistry, tissue or cell can be fixed by the immersion in a buffer solution of aldehyde using the same method that are used for the epoxy resin embedding. Now, the main problem when the fixing tissue is the slow rate of diffusion of the chemicals fixative through the specimen. When possible, it is best that perfusion methods are performed. This may vary from whole body perfusion, where the fixative is pumped through the vascular system of deeply within the animal, to perfusion of isolated organs. When perfusion fixation is not possible, then small tissue pieces should be carefully excised and immersed in a fixative as soon as possible. Okay. So, cutting the tissue into a small piece while in fixative will assist with penetration of the fixative. So, those are the common things that we should all follow. So, sometime that we should dissect the sample properly, tissue properly. So, that should not damage the any area of the tissue. So, that is one of the most important part when you will do the tissue sectioning or handling those tissues. Now, we will go to the actual position where we can see that how we can do the tissue sectioning, or here we are talking about the chemical fixative, as well as we are talking about that at cryo condition how we can fix a tissue or cell and we can do the sectioning. So, here this is the holder which is holding the sample, and in this particular block we want to do the sectioning. Before we want to do that, how we can do a automated trimming of that tissue sample. Here this is a method that I already discussed about, that if this is the blade and this is the tissue sample, we should move at the beginning at the center position of the tissue, we will do some sectioning, and then we make the surface is very smooth. When the surface is very smooth, then we will rotate the blade in one of the side, in this particular side, and use this particular edge of the blade to sharpen the one side of the tissue. So, at beginning we sharpen the front part of the tissue, and this tissue surface should be very shiny. Then we move to one particular side and do some sectioning and polish this particular side. Then we will move that blade to the other side and polish the other side of the tissue. And same way here we will rotate the tissue in the next stage, because we already sharpened one of this edge and the other edge, then we have the two other side that we have to sharpen. So, here we will rotate that tissue to 90 degree and then target the other side and use of this side of the blade to remove the irregular surface and make that smooth. And the similar way we will approach here, we will move that blade to this direction. Now, blade is coming here, and then here we are just using the glass knife, we are not talking about blade, it is the glass knife. Will this glass knife will sharpen this side of the cell, and finally our entire block is ready. Then we will rotate that tissue sample again to elongated version, then we will use this glass knife and gradually get the section of this cell sample. Next, when we will get this section of this cell, there is a loop, this is a tool, that tool we have a loop, and this entire sample, this section will float on top of the water, and then using that loop we will just gently pick up this section and place this section on top of that grid. So, this is the loop we are using to pick up this sample and place this entire section with the solution on top of that grid. Then extra buffer solution, here you can see there is an extra buffer solution. Extra buffer solution will just blot it, and then we will use some kind of staining solution to stain that sample. When staining is done with uranyl acetate or uranyl format, then we can use this sample for imaging. So, there are two images are there where you can see these are showing different kind of cell sectioning and different position of the cell, and there is another example also where you can see different kind of vesicle. This is a cryo sectioning. In this cryo sectioning this cell, you can see inside that there are lots of vesicle inside that cell, which is using that cryo ultra microtome we did the sectioning and we are representing those images. So, this is how we should do the cryo sectioning also to visualize inside the cell. So, in this particular method what we can do, we can image anything inside the cell, it may be the mammalian cell or the bacterial cell, different organelles inside the cell, and we can use room temperature temp as well as the cryo-EM imaging to visualize those kind of organelles inside the cell, and that is the only methods available if we want to see inside the cell. Nowadays there is a instrument called Fib milling. That Fib milling is helping to remove the outer membrane or outer part of the cell, and we can focus inside the cell. So, this is a very advanced equipment that in the later stage, if we get enough time, we will discuss about that techniques also. Thank you all for your attention. In the next class, we will discuss about the cryo-EM sample preparation.